So continuing on with our lecture on metabolism. So last time, we discussed about some terms, endergonic, exergonic, anabolism, and catabolism, as well as the enzymes. Now we will look into some specific examples. So let's look at energy production principles. So metabolism depends on transformation of energy from one form to another. So there's a large amount of diversity of metabolic pathways, especially in the realm of the bacteria. They basically they have almost chemical reaction in most every type of substrate that you can imagine. So they can actually process sulfates. They can process actually a large amount of molecules. They can use it to produce energy, to create their building blocks, etc., etc. Some core pathways are common to most life forms, and that is where we will focus the lecture. We will be focusing on this lecture. So let's see about the oxidation-reduction reactions. So oxidation reaction is when a chemical bond is broken and an electron is lost. While we have your reduction, the freed electron can be acquired by another molecule in a reduction reaction. So basically, redox reaction or the oxidation reduction reactions are always a pair, where in one reaction there is an electron that is lost, and that lost electron is transferred to another set of reaction where there is a reduction occurring. So redox reactions allow energy from one molecule to be transferred to another molecule, such as NAD+, or nicotinamide adenine dinucleotide plus. Basically, it has a charge. So it's a coenzyme that often participates in a redox reaction. You will see a lot of that in most metabolic pathways. So this is an example of an redox or oxidation reduction reaction for NAD+. So we have here your NAD+ in its oxidized form, and it undergoes reduction where it accepts 2 electrons plus a proton to form your NADH, the reduced form. And then when it undergoes oxidation, it loses that 2 electrons, becomes the oxidized form. So one of the mnemonics for oxidation reduction equations is Leo-Ger, or Leo says Ger. Lose electrons, that's oxidized, gain electrons, that is reduced. So Leo-Ger. Actually, there are a lot of them, but Leo-Ger is one of the more common or one of the more famous mnemonics for oxidation reduction reactions. So we were discussing ATP a lot in the previous part of this lecture, the previous video. But what is ATP? So ATP is a high energy molecule that acts as an energy source for most endergonic reactions. So hydrolysis of ATP yields an energy equivalent of 7.3 kilocalories per mole, and your phosphate group. So you have ATP adenosine triphosphate. That is what ATP means. So as from its name itself, it has 3 phosphates. So what is being hydrolyzed there are the phosphates. So you release one phosphate, you release 7.3 kilocalories per mole of energy. So basically, ATP is made up of adenine, ribose, and the 3 phosphate group. That's why it's called adenosine. Adenosine is adenine plus ribose. And then the phosphate groups is the triphosphates. That's why it's the TP. Adenosine triphosphate. So the phosphate groups here can be removed one at a time. So when ATP is hydrolyzed, you form ADP, adenosine diphosphate. So here is your adenosine. So this is the adenine, this is your ribose. So let's look at it, zoom it in. So you have adenine, you have ribose here. So both of them comprises what you call adenosine. And this is your adenosine monophosphate if it's just one phosphate attached. If it's 2 phosphates, it's adenosine diphosphates. And if it's 3, it's adenosine triphosphates. So we do not have adenosine tetraphosphates. We just have up until ATP, adenosine triphosphates. By the way, adenosine is a type of a nucleotide. It's the same as what you have in your DNA, one of the building blocks in your DNA. And this is it's not DNA, it's actually the RNA. It's a ribose, not the oxyribose. So it's an RNA. So you have here adenosine triphosphate plus water, it's being hydrolyzed. So, when you say hydrolysis, you cut it. Lysis means cutting or cleaving away. And then hydro means water. So, you basically cut with water. So, water is being reacting here to remove one of the phosphate groups. So, you have a phosphate, a free phosphate, and your adenosine diphosphate. So, you need to review a bit about your organic chemistry regarding this one. So, this is adenosine diphosphate plus in the cleaving or in the hydrolysis of this molecule, you also release energy. So, that means this is an exergonic reaction. So, why is ATP important? ATP is basically the currency, the energy currency of the cell. Just like how we have the money, the currency for our in buying things, we have money, peso as our currency. So the cells have their own currency. In order to transact reactions, they need to spend or even earn ATP. So ATP production, we have several ways to earn ATP or rather to produce ATP, because again, this is how cells transact with energy. So, one of the ways to produce ATP is substrate level phosphorylation. And in this case, you have an enzyme that transfers a phosphate group to ADP in a coupled reaction. So, basically, it's just simple direct transfer. So, you have here, for example, phosphoenol pyruvate with a phosphate group. And then, this enzyme 1 transfers this phosphate group to your ADP. So the loss of the phosphate of this pyruvate, the phosphoenol pyruvate, will power the addition of the third phosphate to ADP to form ATP. So here is enzyme 1. This one is an exergonic reaction. So this is one important thing about substrate level phosphorylation is that this reaction, the one that will be a hydrolytic reaction, the one that will power the production of ADP, it must have a higher energy release than the one that produces the ATP. So this exergonic reaction, phosphoenol pyruvate, the hydrolysis of phosphoenol pyruvate into pyruvate and phosphate, it's an exergonic reaction that releases 14.3 kilocalories per mole of energy. And the endergonic reaction here, one is coupled. So the energy here is being used to add this phosphate group to the ADP. So this phosphate here will be added to ADP, and this reaction, because you are adding phosphate to ADP to form ATP, this is an endergonic reaction, and this one requires 7.3 kilocalories per mole of energy. So that means this is a coupled reaction where you have an exergonic reaction releasing a greater amount of energy than the endergonic reaction it is used to power. So these coupled reactions has a net endergonic reaction that releases 7 kilocalories per mole of energy. So basically here, what happens in the substrate level phosphorylation is that you transfer phosphate group from phosphoenol pyruvate to ADP to form ATP. And in doing so, you are basically producing the high energy molecule ATP. Now, if you are going to ask, why do we have to do this when you have the phosphoenol pyruvate as a high energy molecule? Because remember, if you cleave the hydrolysis of phosphoenol pyruvate here is 14.3, it's a significantly higher energy release than the hydrolysis of ATP, which is just 7.3 kilocalories per mole. So why? So basically here, ATP is a lot smaller, and it can be used. ATP is basically it's an all-in-one adapter. So it can be used by almost many different enzymes to power the reaction, whereas phosphoenol pyruvate, these are just limited to the specific enzyme that utilizes it. So the enzymes in the body, they are programmed to utilize ATP and not phosphoenol pyruvate. That's one of the reasons. Another reason is that the energy of phosphoenol pyruvate is too high. Some reactions just require 7.3 kilocalories per mole. And if you use phosphoenol pyruvate, it's just too high. It will release a large amount of energy, and the excess energy is usually heat. And then what happens? If you release too much heat as an excess of the energy, you will have, of course, the cell will overheat, the temperature will increase, and of course, uh, there will be many difficulties inside the cell. That means there's a possibility that there might be reactions, unwanted reactions that may occur, or your cells, because of the high temperature, your cells itself will die. So basically, those are some of the reasons why we do not use phosphoenol pyruvate to power anything, but we use ATP. So another way to produce ATP is through oxidative phosphorylation. So oxidative phosphorylation, the energy stored in an activated carrier molecule is used to phosphorylate ADP to produce ATP. This one is for mass production of ATP. So this is the core process in cellular respiration, in particular the aerobic respiration. So we have here a specialized enzyme, the ATP synthase. It's the enzyme responsible for ATP production during cellular respiration. So these are the ways we produce ATP. So in the next part, we will look into actual glycolytic process, actually glucose oxidation. So that's for the next part of this lecture.